Venomous animals have evolved specialized adaptations to deliver toxic substances, often referred to as venoms, which serve various functions such as predation, defense, and competition. The lethality of these venoms varies greatly among species, and it is essential to quantify their toxic potential to understand their ecological roles, as well as for medical treatment of envenomations. The LD50, lethal dose for 50% of the population, is a fundamental metric used to assess venom toxicity. Today, we'll explore the significance of the LD50 venom scale, as well as examine the most toxic venomous creatures across multiple types of animal species. Several factors influence the lethality of venom, making it a complex and multifaceted phenomenon. The composition of venom varies significantly between species. Venoms can contain a wide range of toxins, including proteins, peptides, enzymes, and small molecules. Each component can have different effects on the prey or predator, affecting the overall venom toxicity. The manner in which venom is delivered can influence its effectiveness. Snakes inject venom through fangs, while spiders use fangs or chelicerae. Some species have specialized structures, such as stingers or harpoons, for venom delivery. The LD50 value can differ depending on the target species. Venomous organisms often have evolved venoms tailored to specific prey or predators. The toxicity of venom may be much higher for their intended target than for other species. The age and size of the victim can also play a role in venom lethality. Smaller individuals may be more vulnerable to venom due to their smaller body size and metabolic rates. Several methods are used to determine the LD50 of venomous organisms. In vivo experiments involve exposing laboratory animals, such as mice, rats, or rabbits, to varying doses of venom. The LD50 is then estimated based on the dose-response curve, where the dose causing death in 50% of the subjects is calculated. In vitro methods involve exposing cells or tissues to venom extracts to measure cytotoxicity. While these assays do not directly estimate the LD50, they provide valuable information about venom components and their effects. For medically relevant venoms, such as snake or scorpion venom, clinical data from envenomated patients can help estimate the LD50. This approach is especially valuable when in vivo experiments are ethically or practically challenging. Knowledge of LD50 values and venom toxicity is critical for developing effective antivenom therapies. Understanding the venom's potency can guide dosing and treatment protocols for envenomated individuals. Venom plays a crucial role in the ecology of venomous organisms. Understanding LD50 values and venom toxicity helps ecologists unravel predator-prey relationships, niche competition, and the impact of venomous species on ecosystems. Now that we have explored what LD50 is and have an understanding of its use in determining lethality of venoms, let's dive into some of the most venomous creatures in the world. The inland taipan, Oxyuranus microlepidotus, often referred to as the fierce snake or small-scaled snake, is a highly venomous snake native to Australia. It is renowned for having the most potent venom of any snake species in the world, making it a subject of fascination and caution among herpetologists and snake enthusiasts. The LD50 toxicity of the inland taipan's venom is incredibly low, estimated to be around 0.025 to 0.03 milligrams per kilogram when tested in laboratory conditions, making it several times more venomous than the venom of the Indian cobra or the coastal taipan. Fortunately, this snake is extremely reclusive and rarely encounters humans in its natural habitat. The inland taipan is primarily found in the arid and semi-arid regions of eastern and central Australia, inhabiting areas such as grasslands, scrublands, and sand dunes. This snake prefers to dwell in burrows and crevices, where it can remain well hidden from potential threats. Due to its cryptic nature and remote habitat, human encounters with the inland taipan are exceedingly rare. When confronted, it is known to be extremely shy and will often try to escape rather than engage in confrontations. As a result, fatalities from inland taipan bites are exceptionally rare, and the snake is not considered a significant threat to humans. However, any bites from this species should be treated as a medical emergency, as its venom can be lethal without prompt medical intervention. Conservation efforts are in place to protect this unique and ecologically important snake, as habitat destruction and illegal collecting pose potential threats to its population. The Brazilian wandering spider, scientifically in the genus Phoneutria, comprises several species found in tropical South America, with the Brazilian wandering spider, Phoneutria nigriventa, being one of the most well-known. This arachnid is notorious for its potent venom and its tendency to wander rather than build a traditional web, which has earned it the wandering moniker. The Brazilian wandering spider possesses one of the highest LD50 toxicities among all spider species. Its venom is a complex cocktail of neurotoxins and cytotoxins, with an estimated LD50 of 0.006 to 1.25 milligrams per kilogram, depending upon the species, when tested in laboratory conditions, making it one of the most venomous spiders on the planet. These spiders are primarily found in the rainforests and tropical regions of South and Central America, including countries like Brazil, Peru, and Venezuela. They are adaptable to various habitats, including forests, grasslands, and human settlements, and they often hide in dark and concealed places such as logs, leaf litter, or shoes. While the Brazilian wandering spider is highly venomous and its bite can be extremely painful and medically significant, it is important to note that fatalities from its bites are exceptionally rare. The spider's aggressive behavior is typically a defensive response, and it will often give warning displays before biting when threatened. Effective antivenom is available for severe envenomations, and prompt medical attention can mitigate the risks associated with a bite. Thus, while it is a highly venomous species, its overall threat level to humans is relatively low, especially in areas where medical facilities are accessible. The box jellyfish, Chironex fleckeri, is one of the world's most venomous marine creatures, known for its potent and potentially deadly stings. It inhabits the warm coastal waters of the Indo-Pacific region, including the northern waters of Australia, the Philippines, Thailand, and the eastern coast of Africa. The box jellyfish gets its name from its cube-shaped, transparent body, which can grow to be quite large, with tentacles that can extend up to several meters in length. These tentacles are armed with thousands of tiny, harpoon-like structures called nematocysts, which are responsible for injecting venom into its prey. The LD50 toxicity of the box jellyfish venom is extremely high, estimated to be around 0.04 to 0.27 milligrams per kilogram when tested in laboratory conditions, making it one of the most venomous marine creatures on Earth. Its venom contains a potent mix of toxins, primarily consisting of cardiotoxins and neurotoxins, which can lead to severe cardiovascular and neurological effects in envenomated individuals. Despite its high venom toxicity, the overall threat level of box jellyfish to humans is influenced by several factors. While their stings can be excruciatingly painful and potentially fatal, fatalities are relatively rare due to prompt medical treatment and the use of vinegar to deactivate nematocysts. However, the danger posed by box jellyfish is still significant, especially in regions where they are prevalent, and swimmers are advised to exercise caution and adhere to safety guidelines when swimming in known box jellyfish habitats. The Maricopa harvester ant, scientifically known as Pogonomyrmex maricopa, is a species of harvester ant native to the southwestern United States, particularly the arid regions of Arizona and California. These ants are recognized for their aggressive behavior when defending their nests and their painful sting. The Maricopa harvester ant colonies can be quite large, with thousands of individuals, and they construct distinctive cone-shaped nests with a central opening. The venom of the Maricopa harvester ant contains a mixture of toxic compounds, including proteins and peptides, which can cause severe pain and discomfort when injected into humans. While the LD50 toxicity of this ant's venom can vary depending on factors like the individual ant's size and the recipient's sensitivity, it is generally considered to be moderately venomous, with an LD50 estimated at around 0.12 milligrams per kilogram when tested in laboratory conditions. The overall threat level to humans from Maricopa harvester ants is moderate. While they are not inherently aggressive and tend to forage for seeds and other plant matter, they can become highly defensive when their nests are disturbed. In such cases, they may swarm and deliver multiple painful stings. For individuals with allergies to insect venom or hypersensitivity, stings from Maricopa harvester ants can pose a more significant risk, potentially leading to severe allergic reactions requiring medical attention. Recent advancements in technology have revolutionized our understanding of venom toxicity. High-throughput proteomics and genomics allow for the identification of venom components more efficiently. Additionally, techniques like mass spectrometry and structural biology have provided insights into the three-dimensional structure of venom toxins, enhancing our understanding of their mechanisms of action. LD50 and venom toxicity are essential parameters for understanding the lethal potential of venoms produced by a wide range of venomous organisms. Factors influencing venom lethality are multifaceted, including venom composition, delivery mechanisms, and target species. Accurate measurement of LD50 values is crucial for medical treatment, ecological studies, and conservation efforts. Advances in research techniques continue to expand our knowledge of venom toxicity, shedding light on the intricate world of venomous biology.